Topic operon concept. An operon is a unit of gene expression and regulation. Francois Jacob and Jacques Monod in 1961 explained that metabolic pathways are regulated as a unit. It includes structural genes and their control elements. Control elements are promoters and operators. Structural genes code for proteins, rRNA and tRNA necessary for all cells. Promoters are signal sequences, they start RNA synthesis. Operators present between promoters and structural genes. Repressor protein binds to operator region. Regulatory genes forms repressor interact with operators. Let's watch the given image. Then lac operon. The operon is switched on by a chemical inducer lactose present in the medium. Lac operon consists of promoter site P, regulatory site I and operator site O. Three structural genes Z, Y and A produce an enzyme. Let's watch the given image in absence of inducer. Let's watch the another image in presence of inducer. Then here the actual function of each gene. So lac Z mainly produce enzyme beta galactosidase and whose function is convert lactose into glucose and galactose, while lac Y gene produce an enzyme permease which enter into lactose in the cell, while lac A gene produce an enzyme trans-acetylase which transfers acetyl group from acetyl coenzyme A to beta galactosidase. Then watch the given video to understand the concept of lac operon.
Of the lac operon was the first genetic regulatory mechanism to be understood clearly. So it has become a foremost example of prokaryotic gene regulation. Now if we look at the term itself, it is made up of two words, lac and operon. Lac meaning lactose, which is a disaccharide sugar present in the milk. The word operon means a group. So basically lac operon is a group of genes that regulates the metabolism of lactose. If you have watched the video on translation and transcription, you know that the information from DNA is copied onto an RNA by the enzyme RNA polymerase. The RNA is then read by ribosomes, which uses this information to make proteins. An operon consists of a group of genes whose information is copied by the RNA polymerase onto a single mRNA, which is then read by the ribosomes and converted into different proteins. This bacteria, the E. coli, is a normal resident bacteria in our gut. So it eats whatever you eat. Most of the time you eat carbohydrate containing foods that are easily broken down into glucose in your gut. This same glucose is used as an energy source by the bacteria in your gut. Glucose is fast and easy to use energy source as it need not to be broken down into simple sugars further. The milk sugar, which is a disaccharide, is made up of two simple sugars, the glucose and the galactose. Now this bacteria usually avoids breaking down lactose because of two important reasons. First, breaking down lactose requires energy which is less efficient than utilizing a simple ready-to-use sugar like glucose, which need not to be broken down any further. And second, glucose is almost always an abundant energy source. So when glucose is available, there is no point in spending energy to break down another sugar to use it for energy. But when glucose is not present, lactose becomes precious. The bacteria has to use it, otherwise it will starve. So the bacteria essentially needs a switch that allows it to change its metabolism depending upon the type of substrate that is available. It would be wasteful to produce enzymes when no lactose were available, or if a preferable energy source such as glucose were available like we discussed. So let's talk a bit more about the structure of lac operon, and that will help you to understand the whole concept in a better way. So this is lac operon. This is basically a small piece of bacterial DNA and consists of a few important genes that code for the enzymes that allows the bacteria to break down lactose. The lac operon can be broadly divided into two important regions: the structural genes and the regulatory portion. The structural genes code for the enzymes, whereas the regulatory region of lac operon controls or regulates these structural genes. The regulatory region of lac operon allows the bacteria to essentially switch on or off the structural genes depending upon whether lactose is available or not. There are three important structural genes in the lac operon that are known as LAC-Z, LAC-Y, and LAC-A. LAC stands for lactose, the sugar for which these genes make the enzymes for, and ZYA are arbitrary alphabets used to denote the names of these genes. The gene LAC-Z codes for the enzyme beta-galactosidase. As you may know by now that the sugar lactose is a disaccharide, meaning it is made up of two simple sugars, glucose and galactose, joined by a glycosidic bond. The enzyme beta-galactosidase breaks the bond between glucose and galactose, thus converting lactose, which is a disaccharide, into two simple sugars. The gene LAC-Y codes for the protein beta-galactoside permease, which is basically a tube-like protein that fits into the cell membrane of the bacteria and allows more and more lactose to be pumped into the cell. That is why it is known as beta-galactoside permease, because it increases the permeability of the cell membrane to beta-galactosides, which includes sugars like lactose. The third structural gene in the lac operon codes for beta-galactoside transacetylase, an enzyme that transfers an acetyl group from acetyl-CoA to beta-galactosides. Its function is not fully understood in the metabolism. So these are the three structural genes of lac operon. The region in blue is a regulatory portion of lac operon, and it is divided into many sub-regions. We have the LAC-I gene, which codes for the protein repressor. We also have a promoter, an operator. We also have a capsid, and at the end of lac operon, we have a terminator. At first, these may seem difficult, but I'll try to explain them in such a manner that you will remember the regulation of lac operon in a better way. So, we have our enzyme RNA polymerase that has to copy the information of three structural genes of lac operon onto an mRNA. Now, RNA polymerases always bind at a site located just behind the structural genes, and this is known as the promoter. Just in front of the promoter, we have our operator, and you can remember that the operator as a switch which can be turned on or off. When it is turned off, the RNA polymerase cannot move forward on the DNA, and hence it cannot read what's written on it, so the genes are not transcribed. When it is turned on, the RNA polymerase on the promoter can now move forward, and the genes ZYA are transcribed into a mRNA, which is then used to make proteins. Now, I remember that while learning this, I always used to forget whether the RNA polymerase binds with the promoter or the operator. Well, you can memorize this by just remembering that promoter starts with P, so it binds with RNA polymerase, which also starts with P. P for promoter and P for polymerase. Now the next thing to understand here is what turns the operator on or off, and this is basically the whole concept of gene regulation in lac operon. Now here comes the role of a gene that is located a few thousand base pairs behind the promoter, and this is known as the LAC-I gene, where I is standing for inducible. So this gene continuously synthesizes a protein that is known as a repressor. The repressor is a claw-like protein which can exist in two different forms. When the repressor attaches its claws to the operator, this is known as the bound state, and this is when the red light is turned on by the operator, meaning it is turned off. The RNA polymerase cannot move forward on the DNA because there is literally a roadblock sitting in its way that prevents it from moving forward on the DNA, and the genes are then not transcribed. And you must also note here that this is usually the normal state of lac operon 90% of the times. In the off position, the lac-I gene continuously synthesizes the repressor protein, which goes on and binds to the operator. The operator becomes a roadblock and the enzyme RNA polymerase cannot move forward, hence shutting off the translation of genes Z, Y, and A. Now, since you don't drink milk all day long and the abundant supply of glucose in your gut, the bacteria doesn't need to turn it on most of the times, which saves a lot of energy for this simple bacteria. Now, one more thing that you should remember here is that the lac-I gene is in a constant state of transcription, meaning it produces the repressor all the time, and it is not like the lac-I gene stops making the repressor and then the lac operon turns on. So the question here is, what turns a lac operon on? And the answer to this question is milk and the sugar lactose that is present in milk. This protein, the repressor, has a very soft spot for milk. It likes milk, and whenever it gets some, it becomes happy. And what I mean by it becomes happy is that it changes its configuration whenever it gets milk. See, like I told you, the repressor is normally in a bound state with the operator. That prevents the RNA polymerase from moving forward on the DNA. But the repressor has a site on its back which recognizes lactose. And when lactose fits into this lactose binding site, the repressor changes its configuration. The repressor releases its claws by which it holds on to the operator, and the operator is now free. When the repressor releases its claws, the green light turns on the operator, and the enzyme RNA polymerase now moves forward on the DNA, transcribing the genes Z, Y, and A. So, what does this actually mean for the bacteria? Well, in the simplest words, this basically just means that the bacteria only produces enzymes for digesting lactose when lactose is actually available and not all the time. When lactose is absent, the repressor attaches to the operator in a bound state, and this makes the whole lac operon turn off. When lactose is present in the cell, it fits into the site on the repressor protein, which releases the repressor from its bound state. This is the on position of the lac operon, in which the genes Z, Y, A are transcribed by RNA polymerase. So this lac-I gene and repressor protein is one control mechanism in lac operon, and the basic function of this system is to turn on the lac operon only when lactose is present. But there is one important catch here, that the presence of lactose is not the only factor required to turn on the lac operon. The other factor that must be met here is the absence of glucose. Glucose must be absent to turn on the operon. Now let's imagine a scenario that you drank milk for breakfast and you also ate some bread. Now there is both glucose and lactose in your system, and the E. coli, they are basically having a dilemma about what to use for energy. Now if we had only one regulatory factor here, that is the presence of lactose, the lac operon would have turned on, but since glucose is also present, the bacteria would have benefited more if it used the glucose for energy and ignored the lactose, since breaking down lactose needs spending energy. So this is basically a no deal. Only and only when glucose is completely absent in the bacteria then looks forward towards using lactose as an energy source. Now, like we discussed earlier, when lactose is present, it binds to the repressor protein, which changes it from bound state to free state. So what happens in this case when both lactose and glucose are present for the bacteria to use? Now, since lactose is present, the repressor protein should ideally change to free state, and the DNA polymerase should be able to move on the DNA doing what it does. But since glucose is also present, this would be wasteful as a more easy to use energy source is already available. So why should the bacteria make enzymes for digesting lactose? Now, this bacteria is very smart, and just for this scenario, it has got a special site on its DNA located just behind the promoter region, and this is known as the cap site or the catabolite activator protein site. Now, what is this cap? The first thing that you should note here is that the cap protein is a friend of the enzyme RNA polymerase. Cap helps the RNA polymerase to bind more effectively with the promoter, thus increasing the rate at which enzyme RNA polymerase transcribes the genes of lac operon and ultimately producing more and more enzymes for digestion of lactose. And the second thing you have to understand here is that the cap protein is not always active, and that is how it gets its name, because this cap protein gets activated by catabolism. That is why it is known as catabolism activated protein. Catabolism here refers to the usage of glucose by the bacteria. When lactose is present for the bacteria to use and glucose is in short supply, the bacteria first uses all of the glucose that is present inside the cell. When this glucose gets low, this is a signal of catabolism to the cell and the cap protein is activated. This is essentially mediated by cyclic AMP. This concept is a little long and requires a deeper discussion, but for the sake of simplicity, you can just remember that whenever the glucose concentration in the cell decreases, the cyclic AMP levels in the cell rises. Glucose concentration and the cyclic AMP levels have an inverse relationship. The raised cyclic AMP levels activate the cap protein, signaling the DNA that glucose has been used now and it is time to make new enzymes for digestion of lactose. The cap protein, when activated, binds to the cap site and helps the RNA polymerase to increase the rate of transcription of Z, Y, and A genes in the lac operon. That is why I said that the cap protein acts like a friend of the enzyme RNA polymerase, because it basically helps the enzyme to attach to the promoter more and increase the rate at which it makes RNA. So this cap regulation mechanism ensures that the other factor required for activation of lac operon, and that is the absence of glucose. Whenever the glucose is present in the cell in high concentrations, the cyclic AMP levels in the cell are low. Low cyclic AMP levels mean an inactive cap protein. Without his friend cap, the RNA polymerase feels lonely and depressed and can't function properly, and this leads to decreased transcription of ZYA genes, leading to lower levels of production of enzymes for digestion of lactose. Now this is very important, as lower levels of cyclic AMP basically signal the cell that glucose is present in the cell to use and there is no need for digestion of